My name is Vasilios Liaskonis, and this is my project on soil microbes and the effects of fertilizers on microbial growth. Soil microbes are one of the most important and diverse set of organisms that help vitalize the ground with all sorts of nutrients to keep a stable growing ground that plants would be able to survive in. There are many types of microorganisms that grow in soil, and it is believed that around 1 billion different types of cells are in just 1 gram of soil, which contains bacteria, fungi, nematodes, algae, and even protozoa. These microorganisms help with decomposing any type of organic matter that is dead and brings all of those nutrients back into the ground for reuse and continue the chain for other organisms to use. Now, sometimes these microbes can be interrupted by many things, like being stepped on by a shoe and being introduced to new foreign microbes, or maybe a large storm could bring in certain microbes from a place that is farther away by being washed down onto new land. These organisms have been through every type of harsh environment and are able to sustain themselves in this wilderness. But one of the biggest ways they can be interrupted is by the use of chemicals like fertilizers or car emissions that get drawn into the soil very easily. The question is, how different is the soil that we have in our yards that we tend to groom so frequently and add things to the soil to help the grass turn green, compared to the soil that is in our forests that we like to walk in? Many people today, including massive industries, have been putting tons of fertilizers and different types of chemicals to make their grass green all year round and maybe produce a higher crop yield for a better profit. Some microorganisms will not be able to live in environments that are always changing and being denatured by pesticides and other horrible chemicals. The usual chemicals that are put in manicured lawns consist of many herbicides, pesticides, and a lot of nitrogen and potassium from fertilizers. These chemicals, they kill off a lot of the natural microbial organisms and could create a less diverse number of microbes. This in turn destroys this nutritional value for the soil for other plants to be put in, and in the end, it's a constant cycle of having to add pesticides in all the time because the microbes responsible for putting the same organic nutrients like nitrogen and potassium into the soil have been killed off. So, to see the difference in amount of microorganisms between the two soils could prove a point as to why not to use chemicals to make your yard look just a bit more greener. And there are many other ways you could treat your lawn to have a healthier soil composition and still have a great looking yard. How much of a difference is the soil microbiota that comes from a trail here in Boone than the soil from a well-groomed yard? And how well could the microorganism grown on plates out of our environment? So the hypothesis is the microorganisms grown from a hiking trail should be way better at surviving and grow more diverse than the microbes found in a well-groomed yard, due to yards being sprayed with many chemicals, of course. If the hypothesis is not correct, the only thing I would probably see as a result is that the amount of nutrients and chemicals added to the soil is actually making the microbes grow better and are just as diverse as the hiking soil. So now we get into the methods. In order to go about with this experiment, I wanted to travel to an area that was used as a trail here in Boone, and that people tend to use to hike, but wasn't used too often so that the microbia wasn't too disturbed in the soil. And I wanted to compare a yard that is outside of my apartment complex that is kept up by the groundskeeper and a company that uses fertilizers to maintain it and have it stay green. This yard is also next to a parking lot, and a lot of the chemicals from the cars tend to be washed away when it rains and collects in this yard almost like a pool. So I used 3 plates to collect soil samples from the manicured lawn and another 3 plates to collect from a trail. The way I collected the soil samples was very easy, actually. I went to the area I deemed fit for experimenting, and using a small knife, I would cut into the soil about half an inch and taking a cotton swab and begin to swab the soil itself, right after wetting the tip with a bottled water. As shown in the picture, I would also go about collecting a sample from the start of the side of the trail and collect a sample every 2 feet, totaling 4 feet from the edge of the trail to the final sample collection. For the lawn, I decided to do the same thing and collect a sample every 2 feet from the side of the pavement outwards, and also swab the area, cut open with 3 fresh cotton tips just like before. And using the cotton swabs, I began to wipe the agar plates in a corner-to-corner fashion, trying to get the plate completely filled. After everything was set up, I would take the plates I streaked and mark them 1 through 6, 1 through 3 being the plates from the trail going out every 2 feet, and 4 through 6 was the manicured lawn, with 4 being the closest to the pavement and 6 being the farthest. I collected all the plates and left them under my bed inside of a sealed Ziploc bag at a constant temperature under my room that was 70 degrees Fahrenheit, and would take a picture of them for the next 3 days to see how much growth would occur. What I decided to do was to actually count how many colonies there were on day 1, which was the day after I plated the soil, and try to see how many different looking colonies were visible to the naked eye. Counting the colonies on day 1 would let me know how different each day will be and how much of a difference the plates will look knowing where each colony was before. And here are my results. The results I found for these plates after looking at them for 3 days was not exactly what I expected, to be honest, but let's go ahead and talk about what I saw. I want to compare plates 1 and 4 to start and show you how different the colonies look from each other. The top 2 plates were on day 1 after plating, and you can already see a difference in growth. The colonies look to be more clustered in plate 4, while plate 1 just seemed to pop up occasionally. The color of the colonies for plate 4 started to change colors as well over the course of the 3 days to a brownish type of color. Plate 1 had a consistent looking color throughout the 3 days that stayed the white color you see. But a major difference noticed was that a stringy type of microbe began to form that wasn't present on the plate 4 lawn soil. Counting each colony on the plates, I got around 1,459 on plate 1 and 2,169 on plate 2. Yeah, I counted every single colony for some reason. So, comparing plates 2 and 5 showed almost similar results and had nearly the same number of colonies compared to the plates that were located on the edge of the trail and the pavement. Plate 2 started with 1,448 colonies, and plate 5 started with 2,087 colonies. Plate 5 had multiple clusters and grew rapidly and almost covered the plate in day 2 with just a few spots left open. While plate 2 was growing, it had a lot of empty spaces in between the colonies. And just like before, the lawn soil from plate 4 continued to get dark brown over time as well. Plate 2 looked like the growth of the colonies was somewhat stalling and that the lawn microbes just seemed to grow all over the place. In the plate perfectly fine though. The trail microbes continued to grow this stringy looking type of colony that would mostly grow on the outside of the plate and the inner part of the plate that looked to be an off-white color. Now, for plates 3 and 6, these would be the farthest from the first point of collection. Plate 3, with the trail microbes, had 1,672 colonies, while plate 6 with the lawn soil had only 1,288, a dramatic decrease compared to plates 4 and 5 with the lawn soil. Even though plate 6 had a low colony count, it still seemed to thrive. The colonies in plate 6 over time began to group up and try to cover most of the plate, but couldn't really get the job done. Plate 3, though, actually did a pretty good job, and the microbes began to cover almost all over the plate, and this plate would have the most cell growth compared to the plates 1 and 2. I wasn't really happy, to tell you the truth, with most of the results. I thought I was going to see a major difference between the two. So I told myself I was going to stop here after 3 days. No, I wasn't going to do that. So what I did was wait an extra 10 days to see how different the plates from the trail soil and the lawn soil was going to be. And I'll tell you right now, I'm very glad I did that. So these were the plates after 10 days. Plates 4, 5, and 6 with the manicured lawn soil had almost no cells after the 10-day period. All of them were basically gone. And to my surprise as well, in plate number 6, 4 worm creatures were crawling around in the plate. I measured them to be around half a centimeter in length, with a white head and an orange-like complexion. These plates were sealed completely in a Ziploc bag and were only ever brought out just to take pictures. All 6 plates were all together. So I wasn't going to think that the worms had anything to do with those microbes because it was only in plate 6 that the worms were located in. Plates 1, 2, and 3 still had cells and basically stayed the same from the last time they had their picture taken. And there was no color change, which was kind of remarkable to me. So in conclusion, what actually happened? Well, first I want to talk about how the plates did the first 3 days of growth. Plates 1 and 2 and 3 with the trail microbes actually impressed me for a minute, but it just wasn't enough to actually see how different these microbes could be grown from the lawn microbes. Yes, they were a different color, and yes, they did have that stringy type of bacteria growing on the outskirts of the plate and towards the middle. But I genuinely thought that the trail microbes were going to outgrow the lawn microbes for the first 3 days, and that it would be quite noticeable. Reason being is because I felt as if the trail microbes, being wilder, would be able to adapt faster to any environment and thrive a bit better. So I was kind of disappointed to see my results for these plates. Now for plates 4 and 5 with the manicured lawn soil, they had pretty good microbial growth, which surprised me because I would have thought that something that uses chemicals would not be able to survive that well without its necessary intake of those outside nutrients to help maintain it. So when I saw plate 6 have low microbial growth on day 1, I was kind of like, okay, maybe farther inside of the lawn tends to need more of those fertilizer chemicals to grow. Because I realized that when it rains, this lawn would pull up, and a lot of the chemicals in the soil could get washed towards the center. I decided this couldn't be it because plate 6 after 3 days still had pretty good growth. So this is when I thought to myself that maybe the microbes still have traces of those fertilizers still in the plate, and they are still using it as energy. And this is when I decided to wait 10 more days to see the results. And boy, was I extremely happy to see that all of those microbes in plate 4, 5, and 6 were gone, kind of. Because I was being kind of happy that they were gone. It meant that the soil in the manicured lawn was actually extremely vulnerable. The lawn used so much fertilizer that the microbes highly depended on it, and if the soil didn't get those outside sources of nutrients, they would not be able to survive. The trail microbes, however, were still going strong, which is exactly what I expected to see coming from the wild. More organic type of organism that didn't depend on any outside source of nutrients. It just took me a lot longer to find out the truth and made me stress quite a bit about it. So Was my hypothesis correct? Yes. Yes, I can accept my hypothesis, being that all the microbes in the lawn soil plate had disappeared and that the trail microbes were still alive, meaning that the microbial was able to keep the nutrients in the soil without the help of outside chemicals. The colonies looked more diverse in the trail soil, which is what I want to point out next, because the more diverse the population is in the ground, the better it is for the soil composition. The diversity is necessary because each microorganism has its own role to play out to support all the other types of organisms. If a certain microorganism is killed off, it would cause a catastrophic effect for the soil because the necessary job, which is to supply nutrients for the plants growing on them, is gone. This is why using fertilizers is harmful to soil composition. It only lets the microorganism that didn't get killed off live for only a period of time. But once there isn't any more of the chemicals that help the microorganism stay alive, it dies because it's not able to sustain itself. So what could I have done different for this experiment? And I was thinking about I don't know exactly. Well, actually this. Well, the first thing I would have done maybe would have been to see what kind of fertilizer was being used by the groundskeeper. It would have helped to know the amount of certain chemicals like nitrate, urea, ammonium sulfate, just to know what kind of chemicals that they were using. These chemicals are frequently used in fertilizers. Urea is one of the most common ones, and a study in North China that used urea type fertilizer showed a significant decrease in microorganism diversity while also having a decrease in plant diversity. I would like to try how different fertilizer compositions also affect microbial diversity. And that is my presentation.